The next presentation by M´Jesús Bullido, who's a professor at the Universidad Autónoma de Madrid, and she has always worked on genetic risk factors for Alzheimer's disease and the potential involvement of viral infection on the pathogeny. And this is the talk of her presentation is already on the...
Thank you, Isabel, for the presentation, and thanks to the organizers for giving me the opportunity to tell you why we are working on the lysosome pathway, how we get there, and why we are interested in infection and oxidative stress. In the beginning, our interest in herpes simplex and viral infections in Alzheimer's started as a play. My former director, Dr. Valdivieso, proposed us to try to demonstrate that the possibility of the viral involvement in the disease was false, and we have been for the last 20 years trying to discard the hypothesis, and we haven't been able yet. So our experimental approach to study to try to find mechanisms involved in the first stages of the disease is very simple. It is based on an easy-to-manage model, which is a human neuroblastoma. And in that neuroblastoma, we try to we simulate possible agents involved in these first stages. Now we are validating some of the data we have found in the neuroblastoma in more physiological models like primary neurons, or very recently iPS-derived cells. So we simulate in this model oxidative stress induced with a chemical agent and other agents, which we think in conjunction with oxidative stress could cause the neurodegeneration that we analyze following several general markers of cell death and other more specific of the Alzheimer's, like APP metabolism or tau phosphorylation. Using transcriptome analysis, we try to go to the mechanisms linking the agents with the neuronal death. All of these results come from a very simple cell model, so to validate, to see if these have something to do with the real disease, we use genetic association and, more recently, other biomarkers, in this case, in patients. One example of how this approach led us to results is this one. It's almost 20 years ago, this work. It was the analysis of oxidative stress in the model in, at the moment, it is no problem. I will have it, no, stop. See, no problem, I think I will. Okay. Analyzing oxidative stress, we found that a gene expression in microarrays revealed us that cholesterol metabolism was the most significantly function induced in the model. We then selected a hydroxymethylglutaryl reductase as the possible mediator of the pathway, and we were able to demonstrate that in the model the absence of the genes protected the cells from oxidative stress-induced death, and in patients, we found that the genotype was associated to Alzheimer's disease. And I was very happy to hear that yesterday that this gene is one of the new peaks in the Spanish GWAS study, that hadn't been reported in previous GWAS from other countries. So this has led to novel statins, which are under development, not by us. So a little bit more about the virus, because it is less accepted as associated to the disease than oxidative stress, for which there are many, many evidences. Herpes simplex and other viruses are possibly linked to neurodegenerative diseases, among them Alzheimer's, because two very basic reasons. They are there, they are in the brain, and as I will show you in short, they are able to cause neurodegeneration. Particularly, herpes simplex 1 goes to the brain via peripheral nervous systems, and it can establish latency both in peripheral and central nervous system, and it can reactivate following stress. There are many epidemiological and experimental evidences of the link, possible links between Alzheimer's and herpes simplex, and the first, okay, this one. There. And the beginning of this story, it is not really the beginning, because the virus was studied before the genetic era in the 80s, 70s. But in this year, Ruth Itzhaki showed these results in which she showed that to have the main risk allele, APOE4, was not enough to cause risk, nor to bear DNA of HSV-1 in the brain, but if they were together, the risk was really high. This work has been partially replicated, not by all the groups, as almost everything relating an Alzheimer's risk. So there are other evidences like that peripheral markers of the disease, for example, seropositivity of IgG or mainly IgM antibodies anti-herpes are linked with the disease, that there are genes which are relevant to the infection by HSV-1 that are that have risk alleles for the disease, both from the candidate or from the GWA studies. And there are experimental evidences, for example, the genome of the virus locates in the brain of patients and models. In the plaques, in models, the APOE4 allele facilitates the neuroinvasion by the virus, and both in cell and animal models, the virus is able to induce pathological markers characteristics of Alzheimer's. This leads to this recent report claiming for the possibility to start clinical studies, I don't find the word, sorry. To start to think on the viruses as possible, as potential points to go to the clinic, and actually there are 2 phase 2 clinical trials dealing with herpes infection. It is just in phase 2. Okay. 2 very recent reports have reinforced this possibility of the involvement of viruses, herpes viruses, not specifically HSV-1, and this is this is a work in which the authors, using multi-scale omics analysis, which I cannot understand very well, go to the conclusion that they consistently found abundance of 2 other herpes viruses, 6 and 7, but also of the latency-associated gene of herpes simplex. This work has the difference compared to the previous ones that it is data-driven. It is not coming from a previous hypothesis. It's like the GWAS analysis, and this is the value according to it has no previous hypothesis, as I told you. And this is a very recent, other very recent report in which experimentally in models it is shown that beta-amyloid is seeded by herpes, and that this seeding protects from neurodegeneration. So the viral hypothesis is now, I think, more active than a few years ago, and we are working on that hypothesis. And for example, we have been able also in our model to demonstrate that the virus induced accumulation of A beta, and both 40 and 42. And when we combine this infection with oxidative stress, I'm going fast because I think I'm being too slow. Okay. And when we combine the infection with oxidative stress, these two effects collaborate, and finally, okay, oxidative stress using pH-sensitive vector. What we see is that oxidative stress impairs the fusion of autophagosomes with lysosomes, in contrast with the, I don't know what happens with, thank you. In contrast with classical inducers of functional autophagy. And our conclusion is that when we put infection and oxidative stress together, this last one provokes an inefficient function. It affects the fusion, and in this way it potentiates the markers associated with the infection, which are several, and that which we summarize here. During these years, we have been able to demonstrate in the model that oxidative stress and herpes simplex virus 1 and also herpes simplex virus 2 act and interact, promoting AD-like neurodegeneration markers, including tau hyperphosphorylation, markers related with APP trafficking, and particularly accumulation of intracellular A-beta, and that this is possibly mediated by an impairment in the autophagy lysosome pathway. And all of these works have been published. And to get insight into what functions mediate the neurodegeneration and the markers induced by these agents, we go then to the expression analysis in microarrays. And in this analysis, we simply compare the infected or non-infected cells subjected to oxidative stress. After the analysis, we get list of genes, which are, these are the numbers of genes induced by oxidative stress in infected cells or in controlled cells. We decided to include also in this study another two models in the lab, which are representing a monogenic form of the disease because they carry the APP Swedish mutation and the corresponding control. Also with the idea of finding if there is any pool of genes common to the infection and mutation model, we concentrated in these few genes which are common, which are induced by oxidative stress in infected cells, in cells bearing the mutation, but not in the corresponding controls. And we found that in this collection of genes, there was a huge and very significant increase of the function lysosome when we annotated the list in the Kyoto Encyclopedia of Genes. Okay? There are other functions, but this is by far the most consistent and heavily modulated, and these are some genes of the specific genes pertaining to this pathway. So, what this result told us is that possibly the enrichment of these genes of the lysosomal pathways in both models suggest that they are possibly relevant. They could be involved both in sporadic and familiar forms of Alzheimer's disease. And as you know, there are also many new evidences of this thing, the lysosome dysfunction is possibly associated to many the neurodegenerative diseases, among them Alzheimer's disease. So we are now concentrated in studying the interplay between the herpes and oxidative stress as the triggering of the lysosomal dysfunction that lead to alterations in the markers of Alzheimer's neurodegeneration. And this is the aim of our current MiNeCo project. It is very clear now that the lysosomal network, the lysosomes are much more than degrading machineries, and that there are many vesicles and a lot of sorry, there are some words that I cannot find. A dense crosstalk between these vesicles that form part of the endocytic or autophagic or pathways mainly. And there are evidences for dysfunctions in many of these of the components of this pathway in AD, and we have also in the models evidences. This is we are preparing this work for publication, and we have also in our model are able to recapitulate some of these alterations reported in Alzheimer's. But all of the things we can get from the models will come from the models. So our main interest is to see if this model represents some aspects of the disease, and to do that, we need to go to the patients. And it is only on the patients where we can make some questions unable to be answered from the models. And from these patients, I want to say here just that this part of the work is heavily dependent on collaborations, that I am very grateful to Cybernet, who allowed us to start this type of collaboration that now have given rise to the HESCO consortia, which I am very proud to be part of. And this collaborative work allows us to perform studies like the ones I summarized very briefly here, because Agustin is talking about genetics very much after me. And we are interested in studying genetic association of lysosomal genes from a candidate of GWA study. And we have started to perform gene-focused first step of the studies, candidate genes, and we have performed the first stage of the analysis using our sample, and also part of the HESCO, particularly the sample coming from Dr. Sanchez Juan and Dr. Calero. And by the moment, and this is preliminary results, as I show you here, we have found that LAMP2 gene we have selected 3 SNPs representing 3 haplotype blocks, and we have found that possibly there is a genetic association which is restricted to men of the 3 haplotype blocks, and that is much higher in ApoE4 carrier men. Okay. We have, of course, we need to validate this in bigger samples. One possibility is that we get to this type of results, and this is also a very recent report, performed in big samples using gene pools representative of the lysosome system, and the authors come to the conclusion that they found a diffuse aggregating of genetic perturbation. That means there is no specific genes responsible of the risk, but there is a pool of small contributions. To go to other type of biomarkers, we have started to get a cohort study to determine markers of infection and of lysosomal alterations using different tissues, different samples of these patients. Particularly, we are interested to study HSV, herpes simplex serology in plasma, as well as lipid-related markers. We have started to try to find, to study lysosomal proteins in neural exosomes, and this is based on a previous report that this is possible, that there is neural exosomes in the plasma and that they have different quantity of lysosomal proteins in patients and controls. And we have also started to study in blood mononuclear cells the total levels of cholesterol with Filipin staining and other markers of the lysosome system. Very recently, we have got permission to extract cerebrospinal fluid samples in which we will try to go to these markers. And just very briefly, one slide, I think. This is very, very preliminary data, but I'm going to the next one. Well, in a previous sample, we showed that oxidative stress measured as by the levels of advanced glycation end products, or reactivation of herpes simplex measured as carrying IgM-specific antibodies, allowed us to differentiate controls from Alzheimer's and from prodromic Alzheimer's in this sense. This is APOE4 as the control. And this was performed in a relatively small sample, and this suggests that there is evidences for oxidative stress and virus infection in the, that we can measure in serum. And very recently, we have been able to get this sample, which is relatively big. Well, these are the persons involved. And in these contrast samples, we also see this, the same result. There is more IgM, herpes simplex virus-specific carriers in Alzheimer's and also in prodromic stages of Alzheimer's. We have to validate this, but I was very, very happy because this sample is relatively high, and because of this sample, we also have lymphocytes, so we will be able to cross biomarkers in plasma, lymphocytes, etc. And I'm going to the last one. Okay, we are also measuring Filipin levels. We are starting all of this analysis, and thank you very much to the Cybernet especially, and to the financial agencies, and to all the people who I have not the time to mention.
Any questions? Boys, you're getting tired. One quick question. For this seeding mechanism of the amyloid, the A-beta, with the viral, whatever, what forms or what parts of the viruses you suspect are doing the seeding?
Wow.
Or contributing to the seeding? Isn't that known?
Yeah. That's a very interesting question. I'm not so sure if I know the answer. I don't know the answer. I don't know if it is seen or not. But there are. I think the most probable answer is that the glycoproteins of the environment. I would say that, but I'm not sure.
Let's talk. Maybe it's a simple question, but how the virus access the brain, And if, according to your data, is there any genetic vulnerability to get this? Thank you.
Okay. The virus uses the retrograde path from the periphery. I don't know who are the... hello. The retrograde pathway of the nervous... yeah? It is known, very well known, because this is this virus is very well characterized, uses this pathway. What is not really clear is, well, the most characterized way of infection is through the oral epithelia in adults, okay? But it has also been reported that it can be transmitted from mother to child to children by the hematogenous route. The final destiny is the same, part of the route is the same, but the beginning and first part of the way to the brain depends on the primary site of infection. And in any case, it follows the retrograde anteroretrograde path. It is well. And genetic vulnerability, well, the most accepted data are the ones in these GWAS conclusions telling that there is a general association. There are genes that are related to infection, not specifically to herpes simplex, okay, but to herpes infection that make the people vulnerable. So not any specific gene vulnerability, but there is. And there is also a nice association with gender, which are the women are more susceptible to herpes than men.